Today we're going to talk about biomechanical analysis of 3D in vitro tissue models using AFM. So many therapeutic agents fail in late-stage clinical trials as promising results in earlier in vitro and animal testing have not translated to larger human clinical studies. So there's a study that says that 54% of drugs fail in clinical trials at stage 3. And another study by Wong et al. Estimated that the probable success rate of oncology drugs was only 3.4%, going from laboratory analysis all the way through into patients. And a lot of these drug failures in clinical trials is due to lack of effectiveness and safety concerns. So there is a need for better preclinical models that mimic cellular architecture and environmental conditions within the human body to obtain more realistic results of the way that therapeutic agents perform within the human body. So improving in vitro cell tissue models would provide more clinically relevant data of the effect of therapeutic agents in vivo. It would reduce the clinical trial failure rate of therapeutic agents. And also it would reduce ethical concerns with reduced animal and human trials. And the requirement for better in vitro models can be seen in journal publications, where over the last 20 years, there has been a dramatic rise in the number of publications that use 3D tissue model cultures. Where 3D tissue models have been used in a variety of applications, from cancer research to stem cell research, and also in drug discovery. So research is transitioning away from just using 2D models for therapeutic agent design and also for drug testing, and is transitioning to using 3D models because it's more clinically relevant. So a 3D model is essentially defined as where cells are allowed to grow or interact with their surroundings in all 3 dimensions. And it's an intermediate step between 2D tissue models and in vivo conditions because it has a more realistic architecture of cells. So there are a number of current 3D in vitro tissue models, such as spheroids, and these are multicellular aggregates which are formed in non-adherent environments. Then you have organoids, which are essentially mini organs, and then we also have tissue structures. However, standard characterization techniques fail to capture the rough nature of these 3D tissue models. So conventional light microscopy and fluorescent microscopy only captures a 2D image. And while confocal can capture a 3D image, it is limited by insufficient light penetration into these tissues due to the high cellular density. And also, we have to consider the biophysical properties of these 3D models due to the 3D structural configuration, because these cells are embedded in an extracellular matrix. So in Swansea University, in the Reproductive Biology and Gynecology Oncology group, it's way too much long of a name, so we're going to call it RBGO. We are really interested in 3D models to develop new therapeutic agents with novel mechanisms of action against ovarian cancer. So ovarian cancer is the 6th most common cause of cancer death in females, where the relative survival rate significantly decreases at which stage of diagnosis a patient is diagnosed at. There's such a high death rate because symptoms are very vague. So patients may feel bloated or feel full after eating very quickly. Therefore, ovarian cancer is commonly detected at a late stage. So much so that 17 to 21% of patients already have metastasis when they're diagnosed with ovarian cancer. Also, patients commonly display resistance to current chemotherapeutic agents, such as the platinum-based cisplatin drug, and this further complicates treatment strategies. Therefore, there is an urgent need for new treatment strategies to combat ovarian cancer metastasis. So in Swansea, to further our mechanistic understanding of ovarian cancer metastasis, to enable the development of new ovarian cancer therapeutics, we have first examined the biomechanical properties of ovarian tissues. So in the graph on the left, we have first examined the effect of fixation on the mechanical properties of ovarian tumor tissues, where we can see that fixation by ethanol increases the Young's modulus of the tissue samples. And on the graphs on the right, we've examined the heterogeneity of these ovarian tumor tissues, where we can see that even within the same tissue sample, there are significant variations in the Young's modulus. Furthermore, we wanted to examine the biomechanical properties of ovarian cancer tissues. So here we compared normal ovarian tissues with high-grade serous cancer tissues, where we've seen that the Young's modulus of the high-grade serous cancer tissues was significantly increased when compared to the normal ovarian tissues. And also we've compared different types of ovarian tumours, where we could see that the Young's modulus depends on what tumour type we are examining. However, all tumour types were significantly higher in Young's modulus when compared to the normal ovarian tumour tissue. So, as I said before, in Swansea University, we're really interested in metastasis in ovarian cancer, especially as 17 to 21% of ovarian cancer patients suffer with metastasis at diagnosis. So we need to understand this process. So in cancer, a common metastasis route is direct extension from the tumour to neighbouring organs. However, in ovarian cancer, it has also a unique method of metastasis. So what happens here is cancer cells are released from the ovarian tumour into the peritoneal cavity, where these cells are then aggregated into multicellular aggregates. These are then transported in the ascites fluid within the peritoneal cavity to the peritoneal lining, where these cells adhere, disaggregate, and then invade through the lining, which leads to metastatic colonization of the peritoneal lining. And also, we've really got to understand that in the peritoneal cavity, these cellular aggregates are exposed to shear stresses, such as gastrointestinal movements, ascites buildup, and diaphragm movements. And because these cellular aggregates are exposed to mechanical forces, it's really important to know the biophysical and mechanical properties of these multicellular aggregates to understand the ovarian cancer metastatic process. So in my research, I use the 3D spheroid model to examine the mechanisms of metastasis in ovarian cancer. So, as I said before, I use a spheroid model, and these are three-dimensional multicellular aggregates which are formed in non-adherent environments.